Hi everyone, I am Alexandre Bruni-Cardoso from the Department of Biochemistry, Institute of Chemistry from the University of São Paulo. And first, I'd like to thank Hernandez and Breno for putting together this nice workshop and also for the invite. And to me, that's a great opportunity to talk about something that we are doing in our lab that I am really proud of. A lot of preliminary data, but promising. And I'm going to talk to you on how the matrix helps the cell to make this decision, this Shakespearean dilemma to proliferate or to become quiescent. So we know a great deal about proliferation. We know that proliferation is crucial for all organisms to grow and for reproduction from the single cell organisms to the more complex and complicated organisms. But we know that proliferation forever is not sustainable. So cells need to stop proliferating. For example, here are E. coli bacteria, and they divide real quick every 20 minutes. But then if they divide in an uncontrolled fashion, they can reach a mass that's incredible. That's 3,000 times, around 3,000 times the mass of our planet. So if they proliferate for 40 hours, only 40 hours, without any control, they're going to be heavier than our planet. So proliferation forever is not sustainable. There's not enough resources in our planet to sustain proliferation. And cells from all organisms in the tree of life, from the more simple to the more complex organisms, stop proliferating at some point in their lives and enter a state termed quiescence. And what is quiescence? From the dictionary, is a state of quiet and inaction. But in cell biology, quiescence is a reversible state of a cell when it is not dividing. Quiescent cell doesn't mean that cell is quiet, doesn't mean that cell is not metabolically active. It is doing something. And I'm going to show you in a bit that quiescent cells are not actually quiet. They're not dividing. That's what characterizes quiescence. And quiescence is really conserved. It is as conserved as proliferation. You know that all organisms proliferate, but as I told you, quiescence is really conserved in the tree of life. And why is that? That occurred during the course of evolution for those microorganisms that were able to stop dividing in unfavorable conditions like starvation, lack of nutrients, and they were able to go to a dormant state and then to wake up when conditions were more favorable. So if I don't sleep, I die. So that's the motto of all organisms. So what about complex multicellular organisms? We know that for microorganisms, lack of nutrients is really important. But what about our cells, for example? They stop growing even in the presence of mitogens, even in the presence of nutrients, a lot of glucose, a lot of amino acids, and still they stop growing. So in complex organisms, quiescence is coupled with differentiation. So cells need to stop proliferating to acquire new functions, to do what they're supposed to do, like contracting, like our skeletal muscle cells, beating, like our heart cells, and transmitting electric impulses, like our neurons. And quiescence is really crucial for many physiological processes, but it is perturbed in cancer. So in cancer, uncontrolled proliferation can be seen as a consequence of loss of quiescence regulation. And quiescence is also crucial for organ size determination and also tissue regeneration. So how do the organs know which size they're going to be? And why, for example, in a symmetric organism like us, why my right hand doesn't grow further than my left hand? So how the organs know when to stop growing? And quiescence regulation is really crucial for that, and that's being depicted in this myth of the Greek mythology of Prometheus. He was punished by Zeus, and who ensured that an eagle would go and eat his liver every day just for his liver to regenerate, and then the eagle would go back there and eat his liver again. So suffering forever. Don't mess with Zeus. And we know a lot about proliferation, but we don't know much about quiescence. For example, it is really easy to spot dividing cells under the microscope, but what about quiescent cells? What are the markers? What regulates quiescence? So these questions we are seeking at answering in our lab. And we know that a cell to proliferate needs to go through a cell cycle that is divided in 4 phases: G1, S, G2, and M phase, and finally cell division. And all the key molecular regulators of the cell cycle are known, and they are really conserved from yeasts, from mammals. But what about quiescence? What if the cell molecularly decides to exit the cell cycle? So we don't know a lot about quiescence. So what regulates quiescence in higher organisms? So that's the main questions, one of the main questions that my lab is trying to answer. And we have this hypothesis that the matrix, but not this matrix, the movie, but the extracellular matrix helps the cell, influences the cell to make this decision to proliferate or to become quiescent. As I told you, our cells stop growing even in the presence of nutrients, mitogens. So there has to be something else that regulates this decision, proliferation, quiescence, and we work on this hypothesis that the matrix, the extracellular matrix, influences the cell to make this decision. And what is the ECM, the extracellular matrix? It is a bunch of large molecules, proteins, and some carbohydrates that are made inside the cell, but they are secreted. They go outside the cell and they stay, of course, outside the cell. And this matrix that's composed of collagen, laminin, fibronectin, and many other molecules not only gives a tissue its physical features, its physical properties like shape and resistance to stresses, to shock, to mechanical stresses, but it is also a source of biochemical and mechanical signals that are sensed by the cells. These signals are sensed mainly via the heterodimers of integrins, these surface receptors, and these signals trigger many signaling pathways that overlap with growth factor regulated pathways. So changing many things inside the cells by changing transcription and regulating several cellular processes like the decision of the cell to proliferate or to stay quiescent. And in our lab, we use the mammary gland as a model to study quiescence proliferation. And also differentiation, because as I told you, differentiation is coupled with quiescence. It's just hard to study only quiescence. And we use the mammary gland as a model because it presents many quiescence proliferation switches during its development. And that goes along with lots of changes in the microenvironment, more specifically in the extracellular matrix. And what is really dramatic during the mammary gland development, and we like to say that the mammary gland is the only organ that continues to develop even in the adult, is that during pregnancy it grows a lot, branches, there's a lot of proliferation, and then it starts making milk, and then after weaning, it goes back to regresses, it undergoes an evolution, and then goes back to approximately its original size. And all this cycle can be repeated again if another pregnancy happens. So the mammary gland is really dynamic, and all of these tissue morphodynamics happens along with a lot of extracellular matrix remodeling. And then the developing mammary gland epithelia is really a clear example of how the ECM regulates proliferation. And then this is the developing mammary epithelium. And then this structure here is the growing tip that's called terminal end bud. And then as you can see, it doesn't have a lot of laminin, but the ducts that are left behind when this tab outgrows, the ducts have a lot of laminin. So laminin is associated with cell cycle arrest, and also it induces cell differentiation. So induces the mammary gland cells to make milk. And then, as you can see here, there's a lot of collagen really close to the terminal end buds. So there's a differential organization and concentration of ECM molecules during the mammary development. And then what we are doing in our lab, we are using a mammary gland cell line, a mouse mammary gland cell line, and then we put these cells in contact with a laminin-rich extracellular matrix. So it is a laminin-rich ECM gel. And then in this condition, the cells stop growing, they become quiescent, and they also make milk. If I also add prolactin, which is a hormone that induces milk production in this system. And then this quiescence and milk production happens via a bidirectional signaling. So the cell changes the matrix, and the matrix changes the cell. We know some players that are involved in this process, but we are far away from understanding the whole context, the whole scenario. So what we are trying to do now, what we are doing is systematically knocking out genes that might regulate this process by using a CRISPR-Cas9 screen. And then the idea is, if I knock out a gene that's crucial for quiescence acquisition, the cell will ignore the signals from laminin and will continue to proliferate. And then for doing that, we have established a multicolor cell model. So this cell expresses a probe for the cell cycle and a probe for cell differentiation towards milk production. And this probe for the cell cycle is called FUCI, and it is a dual color probe that becomes red during G1 and becomes green from S to N phase. And then really in a really short window, the probe becomes yellow because of the merging of the two colors. And then if the cell differentiates, and that happens during G1, it is going to become blue because it expresses a beta casein promoter driving the expression of CFP. So if there is stimuli for milk production, the cell is going to become blue. in such a way that I can sort out the cells according to their states. So proliferating cells are those which are green and yellow, quiescent red, and quiescent milk producing are the red and blue cells. And then here, just to show you that these cells really work very well in a control situation, you can see most of the cells are green, many cells are yellow, and then fewer cells are red because they're going through G1, they are actively proliferating. But then when I culture the cells in the presence of laminin and also prolactin, most cells become quiescent, like 95% of the cells become quiescent, and many of these quiescent cells, around 60%, become blue because they are also expressing beta-casein. And then, as I told you, we are using a CRISPR-Cas9 screen. That's a genome-wide CRISPR-Cas9 pooled library. So it is genome-wide because it has 6 guide RNAs for each coding gene, and also 4 guide RNAs for each known microRNA. And it is a pooled library because that's differently from those screens that we are used to, where you have just, a single oligonucleotide or a single drug in a well, and then you need like robots to pipette all your drugs and all oligonucleotides. But here we have everything in the same solution. So that's why that's a pooled library. So we have around 130,000 guide RNAs in the same solution, and then we hit the cells with these guide RNAs. And the guide RNAs are inserted in lentivirus, allowing integration into the genome. And then we make sure that only a single, not more than one guide RNA enters a cell. Hence, not more than one gene is knockouted per cell. So we use a multiplicity of infection, an MOI of 0.3. And then we separate the cells in two, one group to establish the baseline, and the other group we treat it with laminin and prolactin. And then we sort out the cells according to their states. And then we do next generation sequencing targeted to the lentiviral vectors that were integrated in the cell's genome. And then we do bioinformatics analysis to identify enrichment of specific guide RNAs. And finally, we're going to validate candidates of genes of interest. So here are the data that we have so far. So firstly, we look at the cells that continue to proliferating despite being exposed to this ECM that induces quiescence, despite being exposed to laminin. And then these cells were the ones that probably had some important quiescent genes that were knocked out, and they are enriched in these single guide RNAs that knock out these possibly these quiescent genes. And then we plot these guide RNAs according to a score and also according to this KO gene rank versus a KO gene rank. And then in green are those genes that we that were chosen, that were actually, they passed a bioinformatics filter, statistical analysis, and many other bioinformatical filters. And we found a list of 310 genes, so a network of genes that probably regulate quiescence. And that's a positive selection. And then to find genes that regulate lactogenesis, we look at guide RNAs depleted in differentiated cells, in those cells that are red and are blue. And then here we found a network of 320 genes. And then for individual targets that we found, I'd like to highlight some. For the quiescent genes, we found P53 and P10. So we got really happy about it because that's a positive control. So meaning that our assay is working really well, because these guys here are known regulators of cell cycle arrest, the induced cell cycle arrest. But then we found also interesting candidates that we are pursuing by doing some functional assays. So Zoan, Nam, and Chuck, we are now validating these candidates. And then for lactogenesis, we also found these usual suspects, estrogen receptor and JAK2. JAK2 is a kinase that's involved in prolactin receptor signaling. So that's really expected. Again, really good. Our assay is working. And we also found some interesting candidates. EGBP5, integrin alpha 5, and FTO that were not commonly implicated in lactogenesis. Lactogenesis is functional differentiation. So now we are validating these candidates here by knocking down and doing these assays, these quiescence and also quiescence differentiation assay. And then hopefully next time I'll have the chance to show you these data. So now another project still related to proliferation quiescence regulation by the matrix. And I'd like to start by saying that the journey, not the destination, matters. So what do I mean by that? If you look at these two guys, they are on a 10-meter platform. They're ready to dive. And then if I take a picture 4 seconds later, you're going to see that the two guys reached the water. But what happens along the way is really different. You can see that this guy here is a professional diver. Well, this guy here is just, messing around, was just having fun. So the message that I want to convey here is that it's really important in cell biology to look at the events as they happen, rather than looking at fixed time points. And usually the cell cycle experiments are done in this way, like by fixed time points. And then we lose a lot of information when we do that. Another thing is that if you look at this movie here, you're going to see that that's a crowd of people walking in a very busy and big city like New York, São Paulo. But then if I look at these people individually, I'm going to learn much more. I know that this guy here is smoking. This guy here is going to put his forearm on his head. And this guy here is talking the phone. Some people are moving to direction of the street. Some people are not moving at all. So what I want to convey here is that most of the studies of the cell cycle are done by means of bulk biochemistry. Just you put the cells in a blender, you mix everybody, and then you lose a lot of information. You lose the information about heterogeneity. And that's really important. That can be crucial to determine some biological process. And another thing is that usually the cell cycle studies are done by means of synchronization, synchronizing the cells in a specific cell phase. And that's done by using some really toxic agents that can make some artifacts in these cells. So by making videos of the cells in cells that are not synchronized, rather than doing bulk biochemistry, we can learn much more. So bulk analysis, lots of information about heterogeneity. And when I do single cell analysis, usually I can see that outliers become discovery. And then I've heard that from many people, many different scientists. And then I go along with this idea that outliers become discovery. And then just another joke, bulk analysis is like eating a bunch of M&Ms. You're not going to taste any individual flavor. And single cell analysis is like eating one by one, and then I'm going to taste the individual flavors. And then that's what we are doing. We are doing time lapses, videos, fluorescence microscopy of cells expressing fluorescence cell cycle probes. And then we monitor these cells for days under the microscope. And then we use a program, a deep learning program that identifies the cells and then tracks the cells as they divide. As you can see here, you're going to see that every division, the daughter cells are going to receive a number, and then the program keeps track of the cell's genealogies in a way that I can study the cell history and then whether the cell history makes an impact in the cell fate. So where do we want to go? It's been shown this phenomenon of persistence of memory. So CDK2 activity at the mitotic exit and molecular events in the previous cell cycle determine cell fate, and that's been shown by Spencer et al. And then Yang et al. So depending on the amount of CDK2 activity in the mother cell, the daughter cell will divide or will become quiescent. So here in this plot are single cell trajectories. And then these cells that have higher CDK2 activity, they right away after anaphase, they start building up CDK2 activity and they go through a new cell cycle, whereas those cells that have low CDK2 activity, they go into quiescence. They do not build up CDK2 activity, they go to quiescence. And then what we are trying to do is, would this also be the case if the stimuli to become quiescent came from the ECM? And in which phase of the cell cycle and under which circumstances cells would be more prone to respond to ECM stimuli to become quiescent? In other words, is there a restriction point for the ECM? And then we are going to do that. And then just before diving into data, let me take a few minutes to explain to you how the cell cycle is regulated by the cyclin-dependent kinases. Actually, I'm going to focus on the G1 entry and G1 to S transition. So we know that the cell cycle is regulated by the CDKs, the cyclin-dependent kinases, and the first CDK to be active is the CDK4/6, and that's activated by the increase in concentration of cyclin D that occurs in response to mitogens and growth factor signaling like MAP kinase. And then CDK4, when binds to cyclin D, is active, and then it phosphorylates the retinoblastoma protein, which is a cell cycle inhibitor. It is also a very famous tumor suppressor. And then once RB is phosphorylated, that does not bind to E2F anymore, and E2F is a transcription factor that regulates the transcription of cyclin A, E, and also CDK1 and CDK2. And now both cyclin A, E, and CDK2 increases, and then now CDK2 is active because it binds to cyclin A, E, and CDK2 phosphorylates, hyperphosphorylates RB. And that now a feedback loop, a positive feedback loop is established, and then the cell can progress the cell cycle, and CDK2 further phosphorylates and activates effectors of S phase, and then the cell cycle can be completed. So CDK4/6 and CDK2 are really crucial for this decision, molecular decision of the cell to proliferate or to become quiescent. And then using a CDK2 activity sensor That's a fluorescent sensor that is progressively phosphorylated by CDK2 during the cell cycle. And then when it is phosphorylated, it is translocated from the nucleus to the cytoplasm in such a way that I can determine CDK2 activity by establishing the cytoplasmic to nuclear ratio of the fluorescent sensor. And that's a video showing a time-lapse movie of the cells expressing these probes. And then you can see that this guy here is going through G1. This guy here is starting the cell cycle. You can see that the probe is more cytoplasmic, so there's more CDK2 activity going on. And then the cell divides, and then it's going to start a new cell cycle. And then, just to show you that these probes work really well in our mouse epithelial cells, mammary epithelial cells. And when cells are treated with laminin, most of their nuclei are going to have the probe. So the probe is much more nuclear, indicating that CDK2 activity is low. And then if you count the number of cycles that these cells go through when they are actively cycling in a control situation, or when they are treated with laminin-rich ECM, you're going to see that the number is much lower. And then here are individual plots of 3 cell genealogies. And I'm looking at CDK2 activity as a function of time. And then what you're seeing here at the peak of activity that marks G2, and then the valleys of the curves are the end of mitosis and also G1. And then you can see that in a cycling condition, when the cells are not treated, this time where CDK2 activity is low is really, really short. And then you can see what you're looking at here is like a mother cell, a daughter cell, a granddaughter cell, a great-granddaughter cell, great-great-granddaughter cell, and so on. So we can track the cells for generations, and then we can ask the question whether the activity of a cell cycle regulator is important for the cell decision to proliferate or not. And then when these cells were treated with laminin, we can see that G1 is really extended. But these cells are not quiescent forever. As you can see, they wake up at some moments. So that really goes along with this notion that quiescence is transient, so that our system is really a quiescence assay, and then that these cells, they just have longer G1 or G0 phases. Then last year, a new probe came about, a probe that integrates also a CDK4/6 activity sensor. So we can measure CDK4/6 and CDK2 activities in the same cells. And the workings of this probe is the same as the workings of the CDK2 sensor. And then what's being shown by Young et al. Is that CDK4 activity builds up, and right away, after a couple of hours, CDK2 activity also increases. So CDK4/6 and CDK2 activity are really coupled when these cells are cycling, and even in the cells that are emerging from quiescence after serum deprivation and mitogen deprivation, and then just by releasing the cells, by adding mitogens, the cells, they build up CDK4 activity, and right away they build up CDK2 activity. And then we made these cell lines expressing CDK4 and CDK2 sensor. I'm saying here just CDK4 because the mammary gland cells do not express CDK6. So I'm going to refer to this sensor only as CDK4. And then that's in a control situation. We can see that
Both sensors are more cytoplasmic, indicating that CDK4 and CDK2 activity are high. And then, but then you can see that really few cells have more CDK4 activity than CDK2 activity. But then when we treat the cells with laminin-rich ECM, in most cells, the probes are nuclear, are in the nuclei, indicating that the activities of both CDK2 and CDK4 are low in these conditions. And that was also true when we treated the cells with, we cultured the cells in high confluences until these cells reach contact inhibition. So that's another model for cellular quiescence. And what these arrows here are showing is that proportionally many more cells in these conditions have high activity of CDK4 and low activity of CDK2. And then we did a lot of quantification in these cells. So that experiment was an endpoint experiment. We actually have done lots of time lapses, but we did not have the time yet to analyze these time lapses. So I'm going to show you data from endpoint experiments after 48 hours of laminin treatment. And then we made a quantification, and that's like a cytometry plot, CDK2 versus CDK4. And then what you can see is that in both laminin-rich ECM and contact inhibition, in average, CDK2 and 4 activities are low. But then in this quadrant here, you can see that proportionally, there's much more cells with CDK4 high and CDK2 low in comparison to the control cell. So what's going on here? And then here, just a plot showing another type of approach, but the message is the same. So those cells that have high CDK4 activity, they have, when treated with laminin and then in contact inhibition, they have a CDK4, CDK2 activity ratio that's high. So what we think is going on here. So if you look at cells that are actively cycling, you're going to see that CDK2 4 activity builds up first, and then CDK2 activity also increases. And then both activities decrease. And then what we are looking at this plot in this quadrant here is probably this area, this time, really in the beginning of G1, where there's high activity of CDK4 and low activity of CDK2. But then in a quiescence inducing context, like laminin and also contact inhibition, cells spend a lot of time with low CDK4 and CDK2 activity, but then they spend a longer time with CDK4 activity high, whereas CDK2 activity is low. That's really, that's a hypothesis. As I told you, we have not done the time lapses already, but that's what our endpoint data are indicating. That's what we have here is possibly a decoupling of CDK4 and CDK2 activity. And that goes actually against what's in the literature, showing that always CDK4 activity and CDK2 activity are coupled. So what we're seeing here is a decoupling of these activities. So many more cells in this quadrant here, many more cells in this stage where CDK4 activity is high and CDK2 activity is low. So how we think the transient CDK decoupling is happening? So we did a bunch of Western blots for cell cycle regulators, and then as you can see here, cyclin D1, CDK4, cyclin E, CDK2, phospho-RB, they all decrease when the cells are treated with laminin. But P27 increases a lot after treatment. And P27 is really an interesting protein because it has this dual function. It is part of cyclin D CDK4 complex. It is always part of this complex. It is crucial for the formation, the assembly of this complex. And it has both inhibitory and activation roles. So when it is just bound to CDK4 and cyclin D, it inhibits activation of CDK4. But when it is phosphorylated by a tyrosine kinase, it changes conformation, and that stops covering the kinase loop of the CDK4, and this loop covers the active site of this kinase. And then when P27 is phosphorylated, now this T loop is free to be phosphorylated, and then now CDK4 is active. But then P27 always inhibits cyclin E CDK2. So when it is bound to CDK2 and cyclin E, that's not phosphorylated, and that inhibits CDK2 activation by phosphorylation. So what we think is happening here. So in cells that are actively cycling, there is not a lot of P27, just enough to be bound to CDK4, cyclin D, and to be phosphorylated and allow activation of CDK4. And there's not enough P27 to bind to CDK2 and cyclin E. And then in a quiescence-inducing context, like laminin-rich ECM and contact inhibition, there's much more P27, but then in some cells, P27 is phosphorylated when bound to CDK4 cyclin D complex, allowing activation of CDK4, and that remains like this, only CDK4 activation and no CDK2 activation for a longer window, and then some CDK2 activity in some cells starts building up, and then the cell can now progress the cell cycle. So biologically, what we think is going on here, so what's the biological relevance of this CDK decoupling, is that the cell might have a larger window of opportunity to still decide to stay quiescent or to proliferate. And what we believe is that there is a restriction point in between activation of CDK4 and activation of CDK2 that's unknown, and that's something that we are excited to explore more. So I'm about to wrap up, and then I'm going to invite you for some binge watching, just to prove to you that what you just saw is happening. So this cell here, they were expressing CDK2 and CDK4 probes, and then right here, we add laminin to these cells, and then as you can see, the sensors are going to go to the nucleus of these cells, and they will stop proliferating. As you can see, the cells, they calm down, they stop proliferating. And then that's really reversible, because these cells here, they were treated for 48 hours with laminin. As you can see, CDK4 and CDK2 activities are low in these cells, but then when I remove the ECM, I digest laminin-rich ECM, you're going to see that the cells wake up. Now you're going to see the probes being translocated to the cell nuclei, and then they grow a lot. So that's really a reversible state. That's really a quiescence assay. And what if I left the cells for really a long period of time, like 5 days, a week? What happens is that the cells start making complex branched structure. Complex branched structures that's really an emerging event in life. Something that happens in many organisms, like swarming in bacteria, like branching in corals, in plants, in lungs, salivary glands, and of course the mammary gland, where these cells were taken from. So what's probably happening here is that these cells, when they are in a control situation, when they have no extracellular matrix, they forget where they come from, and they continue to grow in a controlled fashion. They do not make these complex structures, but then when we put these cells in their context, which is the context of the matrix, they have a memory. They remember where they come from. When I say a memory, that's an epigenetic memory that we are interested in studying. And then now they make these complex branched structures. So I'd like to finish by thanking especially these two brilliant scientists, Mayara, who's a PhD candidate, and Rebecca Tomazin, a postdoc in the lab. They both conducted the CRISPR-Cas9 screens, and Rebecca is responsible for all the single cell tracking, all the beautiful movies and images that I showed you. These scientists are amazing. I'm really proud of them. And I also would like to thank all my lab members. These guys are heroes. They are really working far from ideal conditions, especially now during the pandemics. And I'd like also to thank Celia Ludio, who's a technician in the laboratory. Without Celia, our laboratory does not function. And I'd like also to thank our many collaborators and many people that shared their lab equipment and reagents with us. Without them, this work was not possible. And this work was initially funded by Serra Pilheira, and our lab is consistently funded by CNPq, CAPES, and FAPESP. So thank you so much. And I know that this video is not going to be live, it's not going to be streamed live, but I'm really open for questions. If you may have questions, let me just go to my email in the first slide here. So you can write to me, brunicar@ik.usp.br. So thank you so much.